Bismillahirrahmanirrahim. Assalamu alaikum. I am here from Conceptual Medicos, and today in this lecture we will be talking about the Haemophilus. Before going to the specific genus, first we should make a background. We have three types of gram-negative rods in the respiratory related to respiratory tract. The first one is Haemophilus, second one is Bordetella, and the third one is Legionella. So the genus Haemophilus, we will take the genus Haemophilus. The genus Haemophilus is further divided into three species: Haemophilus influenza, Haemophilus parainfluenza, and Haemophilus ducreyi. We will be specifically talking about Haemophilus influenza in this lecture. So coming towards the features, so they are small coccobacilli organisms. Coccobacilli means their shape lies somewhat between the cocci and the bacilli. It means that they are small rod shaped like this. Neither they are fully circular, neither they are fully rod. They are encapsulated organisms, which means that they have a polysaccharide capsules. And on the basis of capsular antigen, they are divided into six types, serotypes from A to F, in which the type B is most important. The reason being, most of the diseases by this organism is caused by the type B containing strain of the Haemophilus influenza. Note that the capsule has polyribitol phosphate and that the growth requires the factor X and factor V. Coming towards the pathogenesis of this organism, so the pathogenesis, we have two virulence factors for pathogenesis. The first one is polysaccharide capsule, and the second one is endotoxins, which is contained by all other gram-negative organisms. Note here this organism don't produce any exotoxin. Okay. Infection is only in the humans. There is no animal reservoir of Haemophilus influenza. The transmission of the organism occurs by airborne droplets to the respiratory tract of the recipient human being. Note that when a human being receives the organism to the respiratory tract, some colonize there and are asymptomatic. So the asymptomatic colonization occurs while other get infection. Okay. For pathogenesis, the first step is attachment. And for attachment, the IgA protease produced by this organism help it. We know that IgA is uh, immunoglobulin, which is mostly found on the mucosal surfaces, and this IgA bound to the pathogen and prevent it from binding to the mucosal surface. So when this IgA protease degrade the IgA, then it will become easily penetrated through the mucous membrane. The next step is bacteremia after attachment. Bacteremia means the organism will go to the blood, and through the blood it will reach to the meninges and will cause inflammation there, which is known as meningitis. Most of the infection occurs in the age between 6 months to 6 years, and note that in the scenarios in the scenarios of Haemophilus influenza, the age has very importance.